Okay. Hopefully, this is the last podcast in which we'll be dealing with DNA replication. And now we're going to get very much out of the general idea of the structure of the molecule and that replication is simply the molecule opens and we're going to put complementary bases, nucleotides, attach them to the open parts of the strand. This is going to deal with a lot of detail about how mechanically exactly that happens. Okay? Now, before we go any further, I do want to remind you of where we stopped at the last podcast, and that was in discussing, I believe, the importance of the anti-parallel nature of the DNA strands. It is running each strand, so this is one DNA molecule, this whole thing, and one side is running upside down relative to the other side, okay? And that is not trivial. This is another component of this that you need to remember hinges on the idea that the sugar, the 5-carbon sugar, the carbons are numbered. We have the 1', the 2', the 3', the 4', whoops, and a 5' carbon to which the phosphate group is attached. We can forget about all the other ones, but we must remember the 3' and the 5' carbon, because it is the 3' carbon that is going to hook up to the phosphate group of the next nucleotide in the chain. So we'd get a covalent bond right there hooking up to the next phosphate group, so essentially hooking up to the 5' carbon end of the molecule there. Okay? Very important. We're going to find out why. Okay? So, how does replication really happen? You must be aware of this team of enzymes. It is not just one. I teach my freshmen just one of them and kind of pretend that it does everything, just so they do get the idea there's an enzyme. But we're going to learn all the nitty-gritty details. Okay? So, in the first step of replication, then, we need to unwind the DNA molecule. Of course, we're used to seeing DNA in this double helix form. We are going to have an enzyme that is going to unwind that for us and thus allow us to open up part of the strand as we see here. So, helicase is the name of the enzyme that unwinds part of the DNA helix. We're not going to just completely unwind the whole thing. We're going to unwind a region of it, as you can see here. So helicase would be an enzyme that you could think of as almost, at least in this illustration, acting as a wedge to go ahead and open this up. I'll show you an animation that shows you a more realistic depiction of it. Okay, so helicase is going to be opening up towards this end, and this end of the replication the forks. Remember, we've got these origins of replication, where replication is starting, which open up, and the more they open up, we might call them replication bubbles, so to speak, and each bubble has a region called the fork. And this is the fork at each end, and we're going to proceed down the fork this way and down the fork that way. Okay? Now, the helicase, the opening DNA strand, is kind of held in place and stabilized by some more proteins called single-stranded binding proteins, and these are illustrated by the little blue dots here and up here. So they're kind of just holding the DNA open. Okay? Alright, yeah, cute joke. Okay, replication. Second step is we are going to now have to build the daughter strand based on the template that we were given. So we're needing to add complementary bases to the existing DNA strand. There is another enzyme that does this. It is called DNA polymerase III, implying that there are two others. This is the third one that was actually discovered. Now, DNA polymerase, ACE, is telling you this is an enzyme, so it's catalyzing this reaction, making it happen at appropriate speed. Polymer is simply telling you it's making the DNA polymer, so its name is kind of logical, which sometimes is not true for things like this. Now, DNA polymerase, as you can see here, is going to come in and it's going to add complementary nucleotides as it moves down the length of the DNA strand. But DNA polymerase III has a problem in the way it operates. Okay? Its problem, so to speak, is that it can only add nucleotides to the 3' end of an existing nucleotide. Okay? That's important. Let's see if we can figure out why. Let's look at this. So there's our DNA. Let me back up a minute. There's your DNA molecule. Okay, so we're going to pretend that helicase has opened this up. So we've opened up the DNA. Okay, but look, there's no nucleotides in there where we have just opened it up. There's nucleotides running down this way and then this way, but there's no nucleotides anywhere in here. Okay, so how can DNA polymerase III add to an existing 3' end, or I like to think of it as a little hook, an existing 3' hook, if there's no nucleotide there? Here's my little sugar molecule. I need a 3' hook. There's my 3' hook. There's nothing here. Not only is there no hook, there's no nucleotide. Okay? That is a real problem. So, what has to happen before DNA polymerase III can do its job is we must create a 3' hook so that it can hang its nucleotides onto that. So, if this is our original template strand, here is a nucleotide hook, if you will. And then, once that hook is in place, DNA polymerase III can go ahead and attach things to, and if you'll notice, this was the 3' carbon right there. Okay, it can go ahead and attach things to that, and then we can proceed on with our replication of that molecule. But of course the obvious question is, where did that hook come from? Who put that there? Okay. The hook is actually something called a primer. Okay, primer meaning it's getting things started. And it actually is not DNA at all. The little hook, the little primer, is a piece of RNA. Okay? And that little piece of RNA is laid down by an enzyme that is named primase. Okay? So that's not too bad of an enzyme name. It's telling us that it's an enzyme that lays down the primer. Okay? Primase is the only enzyme that can add nucleotides to the original template strand of the DNA molecule without a hook being in place. Okay, so it is going to be the thing that puts in place the hook needed for the DNA polymerase III to come in and do its job. So there is the hook right there, and please remember, this is an RNA nucleotide. This is not DNA, it's RNA. But once that hook is there, then we are going to be able to add the DNA nucleotides to that. So, the reason that it must work this way has to do with the way we energize the adding of nucleotides. Remember, we are building a polymer from a bunch of little monomers. That is a building exercise, just like building a Lego tower takes energy to do that. We are building something big from small subunits. We need to have energy to do that. The way each reaction is energized, in which we add a nucleotide to the existing DNA molecule, is the reason why we need the 3' hook. Okay? So, where does this energy for bonding a nucleotide to the DNA strand come from when we are doing replication? Alright, let's remember what ATP is. Here's a molecule of ATP. It is basically a modified adenine nucleotide. Okay? So, if we break off that, of course you already know, some energy is going to be released and we're going to get something called ADP. Okay? So, there are other ways to get energy from ATP though. Remember, there's another bond that is also under stress. This bond is under a lot of stress because all of these things are negatively charged and they repel each other, so you flick that one off and it comes off and we can release energy very easily. But these two here are also negatives and negatives, and so if you break the bond there, that's going to release some energy too. Okay? So, but that would not leave us with ADP. If we break things off here, it leaves us with AMP, and if you look at that, why, that's nothing more than a nucleotide. There's your phosphate, your sugar, and your adenine, your base, and we lose these two phosphates, okay? And we release energy when we do that, okay? That energy is going to be used to bond this to the DNA polymer. So basically what I'm trying to tell you is nucleotides, each of them comes with their own energy for their own bonding. It's really quite clever. They can energize their own bonding to create DNA. Okay? So, let me see if I can get rid of all the marks that I have made. Nope. Sorry. There we go. Erase all ink on slide. Alright. Now, so, look, here's a molecule with a guanine instead of an adenine. It's called GTP, but if we cleave off those two, we release energy and we are left with a regular guanine nucleotide. So there you go. It's called GMP in this case, but that energy that's released is going to be used to bond it to the DNA molecule. Okay, same here, TTP, thymine triphosphate, we break off those phosphates, we release energy. CTP, cytosine triphosphate, we break off those, and it's CMP. Okay, so the nucleotides that are going to be bound up to the DNA arrive to the scene as nucleosides. Basically, these are the DNA bases with the 3 phosphates, not just one. Okay, and there they all are. Okay, that's how they come to get added to the DNA molecule. Okay, they arrive with their own energy source, and DNA polymerase is the thing that's going to grab each of these as it's needed and try to attach it to the DNA molecule. Okay, now we can only add nucleotides to the 3' end of a growing strand. Okay, so there's our primer up top, and it's going to grow the strand into the from 5' to 3'. In other words, we're going the exposed 5' is here, and we're going to grow in the 3' direction, and we're bringing our own energy to do that. Now, here's what DNA polymerase does. It's going to cleave here and leave this nucleotide attached to that 3' end. Okay, and there go those little phosphates, and energy is used to do that. The energy released from breaking those phosphates. We're going to do it again. DNA polymerase is going to cleave that. Energy is released. We are bonded again. And here's how we keep going, we keep going, we keep going, we keep going, okay? And that's how we do it. Now, let's look and see if we can figure out what happens if we try to do it the other way. Let me give you one bit of some vocabulary here. What we just did, let me go back, making the strand going in this direction happens very smoothly, as you saw, okay? You just keep adding, keep adding, keep adding, keep adding. The DNA polymerase cleaves off the 2 phosphates and you're good to go. Because of that, this is called continuous synthesis, okay? And the strand that we are making is referred to as the leading strand. Now we'll see why that is in a minute, but it's continuous because it all happens very smoothly. And so what we're looking at here is this side of the replication fork, okay? This side, it's the leading or continuous strand of DNA, but we have got to do the other side, which is going to be a little difficult. On the other side of the replication fork, remember, it is arranged upside down relative to the first side we looked at. Okay, so let's open up. Let me go back. Here we've got our DNA strand. Okay, I'm going to open it up, break the hydrogen bonds here, and we're going to open up the DNA strand. Now, I'm highlighting for you the ends. We've got a 5' end, 3' end, and if the 5' end is on this side because they're anti-parallel, the 3' end is over here, and then again the 5' end on this side is down low. Now, if we are going to add its complement strand then to here, then this is going to be upside down relative to this one, so the 5' end is going to be here and the 3' end is going to be down here. Okay, now we need a primer to come in to add the RNA base so that we can go ahead and hang our hooks onto this 3' thing here. So this is an RNA primer right there that Primase came and added on because it's the only thing that can add nucleotides with no hook at all. Okay, now DNA polymerase 3 comes on in and adds the rest of these in there, no problem. This is continuous synthesis and the strand is growing up into the replication fork with no problem. However, on this side, here's the 3' and of course down low will be the 5' end, but again there's no hook here yet, so again Primase would need to come in and add a hook. So let's add a hook in the same position we did as over here. But what you'll notice is there's no 3' end. What's here is the 5' end here, and that's a phosphate hanging there. Well, let's look what happens. Let's try to add to that. We can't, because where we add is where we cleave here. I'm not going to make a bond there. This is not going to work. Okay? If I cleave here, nothing's happening here. There's nothing to hook onto, and here there's no action happening there. I'm not creating a bond. Okay? So energy is released, but no bond is made. Okay? So what we have to do instead is this. Let me again erase my marks. Okay? Oh dear, sorry about that, folks. Had an itch. What we're going to do this time is we are going to put the primer here rather than starting at one end. We're going to kind of put it in the middle. Now, what's going to happen is, where's my 3' end? It's right there. We can add to that. So instead of growing the strand this way, up into the replication fork, we are going to grow it out this way. Okay? And there you go. We can add here. We can add here. Okay? And if I want to go ahead and do this part, then I'm going to have to add another primer back here or wait for my replication fork to open up more as this leading strand opens up more and more, helicase unwinds more and more. I'm going to have to wait to then add my other primer here, and then I can hang off of that. Now, notice this though does not have a a bond right there. That's a problem. There is another enzyme called ligase that will come in and link those two pieces so that they are connected. So we have a covalent bond, and it is a seamless stretch of DNA. So again, one side can be copied seemingly very easily. Just put your primer in and run it right on up the replication fork. This side, though, a little more complex. We've got to wait for that replication fork to open a bit, put in a primer, and work out. Wait for it to open a bit more, put in a primer, work out. Then we've got to link them up. I do want you to know that the little pieces that the lagging strand, which is what this is called, let me go back, this side over here is the lagging strand, because this kind of leapfrogging, herky-jerky kind of motion takes longer, it lags, than the leading strand. This is the leading strand over here, and it goes a little quicker. The fragments of the lagging strand are referred to as Okazaki fragments. They're short fragments, and remember, an enzyme has to come back and kind of knit those together, and that's ligase. Okay, this is a nice image of showing the leading strand being smoothly replicated as the replication fork opens. We're going up in there. Okay, the lagging strand is coming out this way and has to be assembled in pieces, and I'm going to show you an even better diagram of this in a minute. So Okazaki fragments are the pieces of the lagging strand that are created. All right, now note that only one primer would be needed for the leading strand. You put your hook in and you just zip right on down. The lagging strand needs several primers, and that you do need to remember that these primers are made of RNA, not DNA. So we can't just leave that in there because it's not right. It's DNA we're trying to make, so we have to take that out and replace it. There is an enzyme that does that. It is called DNA polymerase 1. It's the first one that was discovered. That's why it's got the number 1. RNA primer then is removed and replaced with DNA. And then remember, ligase is the thing that is going to stitch together the two fragments once that's complete. All right, I think we can live without that slide. This is the good one that I wanted you to see. All right, and I think this is a diagram on the handout that goes with this podcast. So if we put in our, let's say we have opened up our DNA we want to replicate. So primase would come in and lay down an RNA nucleotide right there. And so the 5' end is going that way, pointing that way. The 3' end of that nucleotide is here, and that's what we need, the 3' hook. And so DNA polymerase 3 is just going to come and very smoothly send it up the replication fork. Over here, though, we have a bit of a greater problem. We have to let the fork open a bit. Primase would come in, lay down RNA primer, and then we would add to that going in this direction. Primer here, we would add to that going in this direction. Primer here, we would add to that going in this direction. And we have to wait for the fork to open more, like maybe right here we could put a primer in and add to that going in this direction, and we have to wait. So we get these pieces, these Okazaki fragments, and these pieces must be knitted together. Okay, right here it looks like DNA polymerase 1 has already come in and taken out the RNA and put in DNA, and now ligase is knitting these two pieces together. That's pretty much a decent diagram of that whole process. Now we're going to do next a recap. So let's do it. There's your replication fork, that your master DNA, your original DNA is opening up. Helicase is doing that. That's the yellow wedge. We've got single-stranded binding proteins that are going to come in and help open that up. These things, remember, stabilize the strand. The helicase is opening it. Okay, let's orient ourselves with 3 prime, 5 prime. So here is a 3 prime end, here's the 5 prime end. So if the 5 prime end of this strand is here, 3 primes down here. If the 3 prime end of this is here, the 5 prime is down here. Okay, now DNA polymerase is 3 would want to go, if this is my 3 prime end, if I were to put an RNA nucleotide here, primase would do that, then the 5 prime would be out here and the 3 prime would be here, and that's exactly what I would want. Okay, so my 3 prime hook is pointing up into the replication fork. Okay, so that's good. So DNA polymerase 3, as long as we've got the RNA primer, can zip right on up there. Okay, but up here, if we put the primer in, the 3 prime is facing that way. Well, there's nothing even to copy that way. There's nothing here. So we are going to have to go, we're going to have to start back further this way if we want to copy something, because we cannot add to that 5 prime end. So what we do instead is we start putting a primer, say, right there. And so if that's 5 prime, this is the primer, the 5 primes here, 3 primes there, we're going to add and move that way. Then we put a little more primer down, 5 prime here, 3 prime here, we're going to move that way, and so on. Okay? Now, so these would be your Okazaki fragments. This is your leading strand, this is your lagging strand. Discontinuous synthesis this way, continuous synthesis that way. Okay, and there's Mr. Okazaki. I'm sure he was a doctor. Remember, ligase would make the final knit. Remember, DNA polymerase 1 would come in and remove the primers and replace them with DNA. Okay, continuous, lagging strand, discontinuous, Okazaki fragments. We've talked about all this. Okay, so looking at this, we're going to see the whole thing. There's your replication bubble, and you can see the 5 prime and 3 prime ends. It's going to open up in both directions. DNA polymerase 3 would move this way very easily up this side of the fork, but up here it's going to have to wait for the fork to open in this direction, but it can do continuous synthesis this way very easily. Okay, so the lagging strand is going to be on opposite sides as we look at both sides of this replication fork. Can you see how that works? Okay, so that's what we're looking at. But just understand why that happens and why this happens. Okay, now, note there's primase, okay, which is going to put down some RNA. Okay, that's what's being indicated by the red. Okay, RNA primer built by primase is serving as the starter sequence. Ligase and DNA polymerase work together to remove those bits of RNA and knit them together. We've already talked about this. This is just showing you that happening again. But what I do want to show you is something right here. Notice this is the primer for that leading strand. But when we go in to fix this and replace it with DNA, notice we take it out because it's RNA, it can't stay there. This is a DNA molecule. But when we take it out, DNA polymerase 1 can't add anything in here because, again, we can't add anything if there is no 3 prime hook to add to. So this creates a bit of a permanent problem. Okay? We can't add there. This is called chromosome erosion. Basically, what happens in our chromosomes over time is that after multiple copying events, okay, we lose bases at the 5' ends of our DNA every time we copy the DNA. Okay? The chromosomes essentially are getting shorter every time we copy them, which means that yes, there is a limit to the number of cell divisions our cells can do because we start eroding the ends of our chromosomes. Okay? Which brings us to the topic of something called telomeres. If you are familiar with your shoelaces, you know there's little plastic covers on the very tip ends of them that keep them from getting frayed. Okay? Well, at the ends of our chromosomes, there are repeating sequences of nucleotides that don't really code for anything, but they are a protective cap, and that's being indicated here by the black. This would be a telomere right there. Okay? And so what that means is we can erode this a bit, fail to copy it, we can erode it, get rid of it, because it's not coding for anything. It's non-coding. It's not giving any instructions for anything. And so we're not going to get damaged. But once we start eating into our DNA that is coding for actual genes and things we need, then we start running into problems, which is there is usually an upward limit of about 50 cell divisions for most cells because our telomeres aren't indefinitely long. Okay, and so there you can see what those telomeres are actually made of. Now, some organisms or some cells actually have something called telomerase, which will replenish the telomeres. Okay, most human cells do not have telomerase, which is why most of our cells have a finite lifetime. However, interestingly, some cancer cells actually are producing this enzyme, which is why they can divide so out of control. In fact, it's thought that our limit to our telomeres is actually kind of a protection against cells dividing willy-nilly in unlimited amounts. So a cell that has telomerase due to a mutation or whatever can potentially be a dangerous thing for us. Okay? Stem cells. Stem cells are cells that can your body is creating in your bone marrow, for example, that can create blood cells that are brand new. Telomerase is active in those because, again, we're trying to create brand new cells. Telomerase is actually active in your reproductive tissue in testes and ovaries because we want the sperm to have completely new telomeres. We don't want to send old telomeres to our offspring. We want to send new ones. So your reproductive cells would have telomerase, but many of our other body cells do not have it, and cancers, as I said, can have it. So a little review. Let's look. If we're going this way with our replication, that's helicase opening us up, single-stranded binding proteins represented here and here. Okay, primase would be laying down primer. DNA polymerase can be seen here. It's been out here already, and it's moving up the replication fork. Okazaki fragments are over here on the lagging strand being created. Okay, ligase is knitting them together, as is DNA polymerase 1 replacing the primers. Leading strand here, lagging strand there. This is just showing you what DNA polymerases look like and letting you know that they go really fast. 1,000 bases every second. 1, there went 1,000 bases. 2, there went 1,000 bases. Amazingly fast, and it is our main DNA builder. DNA polymerase 1 is not quite so fast, and it is primarily an editing, a repair, this is something that can do some proofreading for us to make sure that everything is correct in the DNA molecule and can fix it if it's not. Okay, and these guys figured this out. All right, father and son. Now, DNA polymerase 3 can be prone to typos because it is going so fast. It can make mistakes. DNA polymerase 1, as I just mentioned, is the thing that comes in and proofreads those mistakes. If there are bases that are mismatched, as like, for example, you can see right there, we have a problem, DNA polymerase 1 can come in and fix that and repair it. So it can remove abnormal bases and it repairs damage in cells throughout life. So the error rate of DNA polymerase 3 is like 1 in 10,000, but whoa, we really improve that when we add in the proofreading ability of DNA polymerase 1. Okay? So very fast and accurate replication in a bacterium, E. coli, less than an hour to copy 5 million base pairs in its one chromosome that it has. Okay. Human cells can copy their 6 billion base pairs in a few hours. So, again, amazingly fast. And remember, humans have those multiple origins of replication bubbles, whereas the E. coli would only have one replication bubble. Okay, extremely accurate due to the proofreading and so on. So very, very good. In reality, replication does not look like the cartoon you just saw. It looks like this electron micrograph, and what this is showing you over here are the locations of the replication bubbles. That's a replication bubble. This is one just getting started. Obviously this one's been going a while. Here's one that's not too old, and this one's probably not too old. Okay? Alright, so that's that, and I know that was long. If you got questions, let me know. Thanks for being such troopers. See you later. Bye.